to all the speakers for some great presentations. Now, the even better part of the morning is the discussion phase. And while we're waiting for questions from the audience, I want to tee up one question, sort of a segue from Martin and Alistair, who asked us to not just contemplate what's gone right. Sashi knows what's coming. But what has gone wrong, and what were the challenges, and where have the stumbles been? So I'd love to reflect that question to the panel, and maybe start with Sashi because of the vigorous head nodding.
Okay. Many challenges, as you all know. I mean, there are different perspectives, I think. So submitters' perspective, biomarker developers' perspective, it takes a lot of resources, and that's oftentimes a challenge. And because the biomarker qualification process takes 2 to 3 years from FDA side itself, people can run out of resources if it's grant money, if you're in academia, and even if you're in a consortia, you have limited time frame and monies. So I think that's one challenge. And from a scientific perspective, it's the data. What you have are, sometimes you have data available, but let's say it's in COPD field, or one of the diseases you pick, and then they may not use the biomarker of your interest in some clinical trials. So that data may be there, but the biomarker data is not available. And in some other cases, the company may have the data, but they may not share it with you. So there is, first of all, availability of data, which may be because of either it's not there, or it's there but it can't be shared. So those are challenges. And again, if there are different types of data, it's like having 2 different Lego sets which have nothing to do with each other, and have half the pieces from one and half from the other, and you're trying to make a structure using that can be challenging. So the data standards, I think, is very important. So data access, data sharing, and then of course the data harmonization. So you need data standards, and as I mentioned in the example, it took them more than a year to just get common terminologies and aggregate data. So that's also another challenge. So CDISC standards is what FDA has been suggesting, recommending, and that use of those standards hopefully will help in aggregation of data to analyze it further. So I'll start with that, and there's a long list, but I think I should let others speak some.
Others on the panel with stumbles, things that have gone wrong that you want to highlight?
Yeah, I think what it's been is trying to reach alignment on what a qualification package looks like, right? What's good enough? And I think, if you look, there's only 6 qualifications, right? So there's not a big learning data set there. But every time that we have an interaction with regulators, or we get a new qualification, we learn a little bit more about how this all comes together. And I think over the last 2 years, I think we've seen, a revolution around the relationship that submitters have with the FDA, and how we're working through qualification.
Yeah, I would like to echo that. I think for us, the challenge was the lack of predictability. So the fact that a sponsor or a consortium will put together a package of data, go in front of FDA, and FDA not feeling comfortable and ask for more. So the goalposts keep changing and moving, rightly so maybe, because the data is not there yet. But that lack of predictability really reflect then on the resources invested in a place where it's not giving the ROI needed. And I think the FDA first effort is the CPIM, where you can have this conversation earlier and really start having a discussion about the evidentiary standard. However, I think there is still room for improvement on really setting down what a qualification package will look like, what are the evidence needed, and then definitely looking into the data standard and the assay validation. But provide that clarity, I think will cut a lot of the time and confusion even further when we have many different companies working together.
I agree, and it's been a learning process for us also at FDA. But the good news is, I think we have added a lot of information on the web page as to what's context of use and what are the documents you need and what do we expect to see in those documents. There's also a checklist now to see if you have all those components in a letter of intent, for example, or a briefing document. So we have tried to add that information to the web page to make it useful to submitters. We also published a paper this year, and there's another one coming in November, so that we can explain that to you guys. And we are having all these workshops for evidentiary standards. So we had one in August with the University of Maryland and Critical Path Institute co-sponsored one. This was evidentiary considerations for two types of biomarkers, enrichment biomarkers and safety biomarkers. We have to start somewhere. So we have experience now with safety biomarkers and enrichment biomarkers. So it's good to see lessons learned and where do we go from there. So it's going to take some time, and we need help from academia, industry, and consortia, of course. We have another meeting coming up, the Brookings meeting with NIH, that's coming up October 27th. That's on biomarker taxonomy. So if we can have common, definitions for different types of biomarkers, that may be a good starting point. And then there's another with FNIH that's coming up next year. It's going to be a 2-day workshop on safety biomarkers and evidentiary standards, considerations, whatever you want to call them at this stage. So a lot of work is in progress, and a lot is happening. My suggestion to you is come to FDA early and talk to us early so that we can help and we can work with you. We think of biomarker qualification as a collaborative process, there where we work with you and try to help. So even with a letter of intent, for example, we say, this doesn't sound feasible, or can you reword this? Can we also nowadays offer alternative wording and discuss it? Come up with something, have a upfront discussion on the context of use, and say, let's come up with something that both sides are comfortable with. So I believe that the communication end is going well, and we are also trying to message it by giving presentations in different workshops and meetings and symposia. So we're trying to spread the word, so to say. Thank you.
So before we get to the audience, just one question in follow-up, sticking to the theme of failure mode. And that's, are there examples, protecting the names of the innocent, obviously, but are there examples of things that didn't get qualified, either from the FDA side or from the consortia side or the industry side? And are there lessons that came out of that are really worth sharing right now?
So far, all the submissions that have come to us in the review stage have been qualified. So there were some in the letter of intent stage that we asked for additional information, and that work is being done. So what we have done is we have reached out to all the submitters who we have not heard from for 6 months or a year and tried to get an update from them. And most of them have said they're working on it, and some are prospective studies, so it takes some time. So far, we have not said no to anybody in that stage. However, when people send in a submission, some of those are not appropriate for biomarker qualification. So we explain, we have a teleconference, and we explain some of them are actually to qualify a test. So what we are doing is qualifying a biomarker, not the test itself, though we need an analytically validated test. So some of those probably need FDA approval or clearance. We send them to CDRH to talk to them. So some of them are not appropriate for biomarker qualification. In other cases, there's no preliminary data, so we suggest that it's somebody's gut feeling that this is a good biomarker, or a publication with N = 5 or 10. We suggest that they get more preliminary data, more additional data, and some of them come back, and some of them may not have come back. So that's, I don't know if it's lessons learned, but just wanted to explain the process there.
Good, good. Go ahead.
Yeah, I think one effort the FDA has done, which helps, I think, is the visibility of what is in the process. So now FDA publish all the biomarker that went through the gate with the letter of intent, and actually show if it's a clinical, non-clinical, a safety versus efficacy, and then also if it's in the letter of intent on the consultation and advice or review, and have point of contact. So I think sharing what's out there and what is, I think it can help the communication among investigators and can definitely help with the data generation. I think when you mentioned, once they get to review, they make it to qualification. I think more that the consultation and advice is where everything gets trapped. And that's where the conversation with the evidentiary standard. So it's like, you go through a lot of boyfriends before you actually get engaged, but when you get engaged, you're gonna get married. So that area right there is the most difficult one. Find the right person, find the right balance between risk and benefit and the evidentiary standard needed to move that qualification forward.
I just wanted to add that we asked all the submitters would they like to share their information on the FDA webpage. So I think 18 or 20 out of 24 at that time said, yes, we would like to share the information. A few people said we don't want to share the information. So we want to respect all the submitters as at this time anyway, it's not mandatory. So, we're trying to share what we had. And yes, a lot of people think that things get stuck in the consultation and advice stage. It's not really stuck. It takes time for the biomarker developers to go back to the drawing board sometimes, or to wait to get access to available data. So let's say an industry has some data, and the lawyers and everybody has to work together and give access to the data. Sometimes it's just a waiting game. And if the data is not there, there isn't very much we can do, and we don't have a deadline. So we wait for the submitters to gather the data that's needed. We talk to them, we give comments saying that, and suggestions as to what we want to see in the next stage. And then we also have for a briefing document, for every briefing document submitted, we give an option to have a face-to-face meeting with us or a teleconference. So we really work with the submitters to get them to a stage where it can go to the review stage. There is no point in taking something to a review stage if the submitters do not have all the data needed. It's wasting everybody's time. So I think it makes sense. The letter of intent is what it is. It's a letter. It goes through quickly to the next stage. But consultation and advice is where the biomarker development really occurs. And sometimes the proposed context of use doesn't pan out. What you have as a starting out context of use is a hypothesis based on your preliminary data. But then when the real data comes in, the context of use may need to be changed, and we are okay with that. We are flexible about that. But sometimes that changed context of use may need some other kind of data. So, it's complicated. But that's why the biomarker development taking more time is what is the delay, the need for delay. If somebody, proposes a context of use that pans out, and they have all the data sets they need, all the ducks in a row, as they say, no matchmaking here, but all the ducks in a row, and then it flows more smoothly. So there are some submissions which move very smoothly, very quickly, and others which take longer time.
So we have a lot of folks standing by the microphone, so let's get to some of the questions from the audience. Russ?
Yeah, thank you for the talk. So very interesting. I wanted to ask about if there's any experience or anticipation of biomarkers that are signatures, in the sense that there are a number of different measurements made, they are combined in some kind of algorithm to give some kind of output, and perhaps none of the individual measurements would qualify or even be of interest, but as an aggregate they become potentially useful. I'm wondering what the issues are there and how that future looks.
So I know exactly where you're going with this. One of the examples that I gave is actually a composite measure. It doesn't quite get to your example because those biomarkers ultimately will be used individually. But you're right, and we have not approached that within the PSTC. Shashi, do you have any examples?
We do accept people submissions that come with signatures. We call them composite biomarker.
Yes, yes.
And it's hopefully a homogeneous composite, which means all biomarkers. But that's totally acceptable. You just have to show data, because the signatures like proteomic signatures or gene expression signatures, those are a panel. You cannot think of those as one entity.
The reason just as a follow-up, I would imagine that the algorithms that do the data mushing together, to use a technical term, that those would also be then the object of interest for you guys. And I'm wondering if if the principles that you already have are perfectly applicable, or if you need to extend your scheme for evaluation when there's these algorithms as part of the biomarker.
We do look at the algorithms for sure, and we haven't so far worked with huge, numbers of biomarkers, but we have worked with multiple biomarkers. And right now, those are not in big numbers, as I mentioned, and there is weightage given to one biomarker, sometimes a little more than the other.
Right.
So all that's considered. And our colleagues from CDRH, we always have one reviewer from CDRH who helps us in evaluating some of those.
So why don't we go to the other side of the room?
A question for Mark. And for all the panelists, I really appreciate the amount of work it takes to validate, standardize, et cetera. But specifically for Mark, one thing I struggle with is sort of the dirty little secret of gene expression measurement is just how variable it is. And the question is, do you really think we're going to standardize our way out of the problem, as opposed to standardizing methods to reduce noise? So as an example, we've done a lot of studies where you compare two systems to the level of individual genes, and the Pearson's R is awful. You then filter that through GSEA analysis, and you find a great correlation between the gene sets that you hit. So it's a noise reduction methodology as opposed to a standardization approach. So there's an example, but the question is, given the many, many sources of variability, platform users, are we ever really going to standardize our way out of the problem using individual genes as measures?
Thanks, Jim. That's a great question. And the short answer is, I don't think we're going to calibrate genome scale measurements to the point where you're going to be able to compare results over space and time, where a panel that I measured on a cohort today would be applicable and completely without doing the kinds of noise reduction that you describe, or some sort of aggregation or evaluation at the pathway level, where it could be used 3 years down the road with incompatible equipment. It may, and we already are so much better than we were a decade ago. So to predict that it's impossible would be silly. But at this point, calibration at the genome scale with a lack of understanding of the interactions seems intractable, or maybe not even that interesting.
I think this is a problem that's completely underappreciated and needs to be elevated to produce solutions.
Yes, and speaking of problems that should be elevated, data quality and access to data often hinges on the ownership of data. What kind of recommendations should we make, and how do the different stakeholders view that, even in the panel? Like pharma and bio probably have a different opinion than FDA has.
So do we want to go to the consortia answer for that?
So, I mean, as I put on that last slide, I think one of the discussions that really needs to happen is really what's pre-competitive now. I mean, we had these discussions as consortia were forming, 8 years ago, somewhere around there, and we decided where that fit. I mean, we really need to look at where we can leverage the data that's being generated in clinical trials, be able to de-identify and mask that data, and be able to then use that for biomarker qualification or for other, solving other issues that we have. we have examples of that within CPATH. We have an Alzheimer's database, which basically pulled together data from multiple companies, and that was something that probably couldn't have been done. It couldn't have been done with a single company, and so I think we need to think a little bit differently now that we've invented drugs for all the easy diseases out there. Now that we're working on the hard ones, I think we need to come up with a slightly different model than what we've been working with in the past.
What do you think that model is?
Well, I think that model's around data sharing. You know, actually figuring out where pharmaceutical companies, where the competitive space is for pharmaceutical companies versus this pre-competitive space. Clearly, all the safety information that's generated is not competitive. I mean, I think Jim did a really nice job in putting that together and explaining that. I think there's other areas as well beyond patient safety where we could do the same thing around either trial enrichment biomarkers or ultimately in biomarkers that will become surrogate endpoints.
And yet, many biomarkers continue to be considered proprietary for different reasons. Sashi just talked about there are folks that didn't want their packages shared, their data shared. So how do we get out of this hole? I'm sorry, I keep looking at you, John Michael.
Oh, no, it's okay. I mean, because if you think about qualification, all that data is going to be made public, right? And everybody's going to be able to use those biomarkers. I mean, if you really want it to be a proprietary item, then it has to be in your IND. But there's risk with that. I mean, qualification brings regulatory certainty. There's a guidance out there. I know how regulators are going to react to my data. Within the IND process, I could be denied even using that biomarker for a given program without, and asked to generate more data. So I think it's a risk-benefit type approach.
My name is John Birch. I'm an angel investor, a member of the Angel Capital Association. I'm based outside of Kansas City, and I'm going to bring up something that I hadn't expected to bring up at this meeting on regulatory science. The area, Kansas and Missouri, is regarded in many areas as the animal health corridor of the country. And I wanted to raise a question really about the non-clinical trials, not the non-clinical space. There are now more and more electronic medical records, if that's the right term, being used by veterinarians. I wonder if that might not, and let me just also say, as an angel investor, in the last year alone, I've heard at least 3 pitches by entrepreneurs who are developing some kind of new intervention for animal health, pets, dogs, dog cancer, for example, a number of things. And they all stress that the FDA, they do have to go through the FDA. They all stress, however, that those thresholds are significantly lower. I haven't really looked into it for efficacy and safety for animal products. But my point really is that I wonder if this doesn't possibly provide an area for testing biomarkers or other efficacy, other matters as well. I know that it's already of some interest to epidemiologists to look at, to keep track of animal health issues, animal health results, because a lot of infectious diseases that start with dogs and cats end up in children and people. And so we can get a kind of an early indication of some infectious diseases that way. That's a completely different matter, but there may be some other uses as well that could be made of the electronic records that are now available from veterinary sciences. Just throw that out. I wonder if any of you have thought of that.
So we've been talking to a couple vet schools actually around the use of these biomarkers that we're working on as the next generation of biomarkers for animals. Because it turns out that the exact same problems and some other ones are associated with veterinary science and the care of the animals. So what we're looking at is, can we apply these biomarkers? They didn't think about the electronic health records are going to that next step. But one thing we also need to think about is many of these safety biomarkers are probably also going to end up to be efficacy biomarkers for different indications. And so there's also that overlap.
Burroughs Welcome Regulatory Science Review Committee, and we received an application actually for a grant that was looking at veterinarian data as a means of looking at efficacy of drugs and other approaches to human disease recently. So people are starting to think about that. Rusty may want to comment on that if he's still in gears.
Another question?
So we only have time for one or two more questions, so if you could.
So I hate to keep dragging John Michael back into the cesspool of the pre-competitive space, and maybe I'll take Gabriella down with you.
It's not a cesspool.
Well, is it? Right. So, do we have a clear idea of what the pre-competitive space is? Who gets to define that space? And then what are the real and the perceived problems? So perception being reality sometimes. Sometimes the perceived problems with the pre-competitive space can be overcome.
Well, thanks for the easy question at the last minute.
No, lawyers. No, I'm teasing. I think well, who needs to define the pre-competitive space? Clearly, it's pharma, right? It's their data, and so the question is where's that risk threshold that they want to go ahead and approach? I think when you talk to pharma scientists, there's this willingness to share the data. You even escalate and go up to the management, and there's a willingness to share data. But then when it goes up beyond that, and we begin talking about the legal issues and the risk associated with that, that's where we run into problems. And I think it's ultimately going to be that from a legal standpoint, pharma needs to figure out, what risks they're willing to take, and thus my answer: no, lawyer.
Or is it fear of loss of competitive advantage? What is the risk? It's a big bucket.
Would you like to take that?
I mean, I think it's all of that. So you have competitive advantage, you have IP. I think many times you cannot just extract one data point. It's like extracting a tree. It has a lot of roots that goes in the proprietary things. So I think there are two pathways, and we support that public process where there is sharing and collaborating, and then there is the IND, NDA, BLA pathway, which is the proprietary pathway, and the sponsor has the right to keep that proprietary. But that does not stop each individual sponsor at their willingness to share data with others. And I think you have a variety of cultures and risk aversion within the industry. So you will find members and companies that are more willing to share, and some they're not. And I think if they are in that proprietary pathway, all that spectrum is allowed. But we still need to protect the company willingness to keep everything private.
I'm sorry, I have to intersect.
So
I'm
I
not
think
a
that's
lawyer,
the secret, right? Is
if I
to
may.
create that environment, if you would, where the sharing can happen, and we need to figure that out. That's the discussion that needs to happen. Maybe not what, but how initially. How do we go ahead and mask and de-identify data so it can't go back to a sponsor and they don't incur that risk?
I think there was
Yes,
one more
one additional comment in this context. I'm not a lawyer, but I would also offer an additional or alternative approach. Pharma conducts trials in human beings at some form, and at some point, human beings are not owned by pharma, so you can go back to the debate who really owns the data. And I think if you look at it as a society, it is critical to understand what the risk is for society in our investments that are made from different contributors and different stakeholders in this context. And I think it would lead perhaps to a different outcome of this discussion as we have had it in the past.
So to that comments or question, FBR and pharma have put together the principle for responsible clinical trial data sharing. So a lot of the members are certified, they obey to those principles, and they receive queries from outside stakeholders to actually see the data. And many of the members look at that and share. It has to be a vehicle and a platform for having that sharing for what and for which reason. But again, it has to be decision of the sponsor itself.
So I will end the session by saying that we've already eaten into 5 minutes of the lunch hour. and so there are certainly many questions that one could talk about regarding the biomarker area, including some that are not necessarily scientific, such as cost and scalability of this approach, et cetera. But I think that we need to break for now, and we plan to be back very sharply at 10 minutes to 1 to begin the next session, which is on clinical trials, and so that could be a follow-up to the very last comment. So are there specific mentions regarding lunch?
There is lunch. And
wasn't given any additional instructions.
And I just wanted to thank the speakers who did a fantastic job.